No, I just got a text. Oh, I'm in now. Hi. All right, bye. Hey guys, sorry I was late. I totally forgot when our class was. This is typical of me. I'm thinking, oh, I've got till 2 o'clock. I've got till 2 o'clock. And I swear to God, I finished the notes at 1:02, I think. So I just barely got ready. Um, but well done. Want to thank, oops, cancel. Want to thank Sam Robinson and Angelique for letting me know how stupid I was, telling me to show up. And so now I'm here. Okay, let's rock on. All right, so I'm going to post the notes right now. So if you want to follow along the notes, you can. Again, sorry I'm so late going on here. I don't know what happens with my mind. It's just like I know, I know I've got class, but why is it suddenly I think it's at 2 when it's always at 1? I don't know. Oh, it ended at 2. I see. I see. Okay. Okay. Actually, I've got a whole lot of new slides I added today too. So I'm deleting the old PowerPoint and I'm uploading the new PowerPoint. Bum, 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 bum. Okay, lecture notes are there. Now, PowerPoint. New PowerPoint is on its way. It takes a little while because it's a big file. Oops, it didn't happen yet. Okay. All right. So let's start this class. I know you're all anxious to learn. So today we're going to finish chapter 3 and talk about some techniques that are used and we'll use these techniques in the future in the coming classes. So when I do mention them again, you'll know what the heck I'm talking about, hopefully. Come on, man. Trying to, oh, come on, man. Trying to change the size of my PowerPoint here for you. And every time I do, the Zoom menu shows up and gets in my way. Anywho, almost there. I promise. I swear to God, people, we are going to have a class today. I swear to God. Unless you want to take today off, let's have a quick vote. By thumbs up, how many people want today off? It's a democracy, right? Give me a thumbs up. Yes, yes. I'm seeing some yeses. I'm seeing some yeses. Yes, yes, yeah. Well, unfortunately, I'm in charge and I say we're going to have a class. So thanks for your vote anyway. Okay, so the last thing we talked about was this in situ hybridization stuff, which I think is really cool. Again, I don't know if I said this last time, but if you are planning to do research for the rest of your life, find some way to include immunofluorescence because it's beautiful. It's some beautiful stuff. So here is nucleic acid hybridization. So you're putting different chromophores on top of DNA and then putting them onto your fixed embryo, and you're seeing where the RNA is for those things. So you can see where those particular genes are turned on. And I think in this one, we've got 3 different probes here. You've got the blue one, you've got the red one, and the green one. And when red and green line up, they turn yellow. So you can see overlaps as well as where they're individually expressed. And this is, I mean, look at this. Why wouldn't you want to spend your life looking at things like that? That's gorgeous. It's gorgeous. Okay, enough of that. And this is how it's done, which we talked about last time. Oh, this is immunofluorescence. Okay, okay, okay. And here we are. So the next thing to talk about is, again, when we're looking through these, when we're looking for at embryos, right? You can look at all the physical structures all you want, but really what we want to know is what genes are being turned on. Because the genes get turned on, they make the protein, and then the protein changes the cell. Most of what we talk about here in developmental biology is about differentiation, changing from one type of cell to a different type of cell. So what proteins are important to cause that change, to make it a different cell? What genes are the transcriptional activators that will turn those genes on? What are the hormones that will turn on the transcriptional activators that will turn those genes on? So really the key to the last probably 40 years of research into embryology has been what genes are causing all this to happen. And that's mostly what we're going to talk about from now on in the class. So how do you know what genes are turned on? One way is by DNA microchips. So this is an example I got from the web about DNA microchips. Basically, you've got a bunch of normal cells and a bunch of cancer cells in this case. You isolate the messenger RNA, you put them together, and you can see which genes are expressed only in one cell versus what genes are expressed in another cell. I'm not sure I'm crazy about this example. I think this is a better way of looking at it. So here's a DNA microchip that you can, I think you can buy these, like mouse microchip. And so every little dot is a piece of a gene, piece of a different gene. So what you do is then take your unfertilized egg, you isolate all the messenger RNA from it, you turn that messenger RNA into DNA. Now, messenger RNA in general is very labile. It breaks down very, very easily. So in order to be able to do some half-decent experiments with it, you take that messenger RNA and you add an enzyme reverse transcriptase, and you make the DNA that's complementary to the RNA. That's cDNA. So you make all this cDNA from the RNA that's in the unfertilized egg. You put that in the microarray. So you see what genes are turned on already in the unfertilized egg. Then you fertilize that egg, you do the same thing. Oh, what genes got turned on when it got fertilized? And so on and so on and so on. So you can follow, and again, by looking at this array, you can see which genes are turned on in which states. And then you can further look into that gene to find out and then do more experiments with that gene, like we'll talk about later today, like how, once you have that gene, what do you do with it? Okay, so that's DNA microarrays, really great way of detecting which genes are turned on and in which developmental states. You can even dissect away different pieces of the embryo and do a DNA microarray to find out, what's being turned on in this specific tissue at this specific time. Do I have another figure on this? Let's see. Okay, I don't. All right, well, anyway. Okay, so that's finding out what proteins are being made, what things are being turned on. The next experimental technique is to find out what control elements what control elements your transcription activators are binding to. So you've got a gene that is binding to DNA. It's a trans factor, a transcription factor. And so you want to find out what genes it's binding to, so you can see what the sequences of that control element. So just a quick foray into gene structure. Here's the protein coding region of the gene, exons, and then introns that we'll cut out later. And this is controlled by the promoter, and then also upstream enhance elements. So this is what we're looking for. What sequences are our protein binding to? So what you do, and here's another look at cis-regulatory region. So the proteins then bind to these upstream regions, and then that causes the DNA to fold over and bind to the proteins on the core promoter, the TATA box, and then transcription will start. So we're looking for these proteins then. So what you do is you take your genomic DNA from your model organism, mouse, chick, whatever, and you add some of your protein that you've made in vitro to that genome, and you let it bind. Just let it sit until it finds its spot and binds to it. And then you hit it with a chemical that will covalently link it to that piece of DNA. So now it's stuck there, it can't get off. And then you cut your genomic DNA up into little pieces. So some of those DNA pieces are going to have your protein bound to it, very few, okay? A protein may only bind to 10 genes. So you'll have just a little bit of DNA that your protein is bound to. But because we know the protein, we can make an antibody against it, and then we can find those pieces of DNA really easily. So you've got your DNA, you add an antibody to it that's going to bind to that protein, and then you run that over a column. Let's see what our next figure looks like. You run that over a column, or you add beads that will stick to that antibody. And now this will immunoprecipitate out. All those other little pieces of DNA are too small to precipitate out of solution if you do your conditions right. But the ones that are all bound up together like this are heavy, they come out. Now you get rid of all the junk and you isolate only this DNA that you're interested in. And then two different ways you can go with it at this point. You could fluorescently label it and do your microchip array to find out what genes that is associated with. Or you could just sequence that DNA and find out what the sequence is. Once you know the sequence, you can go to the mouse genome bank and find out what genes are have these cis regulatory elements. And therefore, your protein probably regulates one of those genes or all of those genes. You never know, but it's just a way of getting information about what genes your transcription factor of interest is going to turn on. Okay. Okay, so I went over that kind of fast. Let me do it again, just so I make sure I did it right. Okay, so it's all about finding these cis regulatory regions. So cis just means it's connected to it. It's the same, it's part of the same gene. They used to talk about cis versus trans in terms of gene activation. Trans is when one gene affects another gene somewhere else on the genome that's not connected that way. And that basically means it's the protein. So the trans factor is the protein. Cis factor is directly connected to the gene. So that's why we call this the cis regulatory element. Cis is the same, trans is different. Okay, so there's your cis regulatory element. And we're binding our protein to it. And we're basically isolating whole genomic DNA from your model organism, binding your protein to it, and then chopping it up in little pieces. And then we can isolate the correct pieces because our protein's bound to them. We get a bead with an antibody on it and an antibody specific for this protein. And then you get these big clumps of protein antibody bead DNA. And then you can get the protein off the DNA and then sequence it or hybridize it to a DNA chip and in that way find out what genes it's labeling. Okay, questions, comments? Not sure we're going to talk about this so much in the next rest of the class. Mainly, I'll tell you what the elements are and you can just kind of in the back of your head say, oh, I bet you they figured it out this way because that's probably how they did it. Okay, now here's something we will talk about a lot in the days to come, and that is fate mapping. So fate mapping means here's your embryo. This is what, 4 times 4, 16. So this is your 16-cell embryo. And of this embryo, you know, it's only got one axis of rotation. So what happens to each of these cells as the thing develops? So what you can do is inject a non-diffusible dye into one of these cells, And because it's not diffusible, it's never going to leave that cell, but the cell will divide and make more cells, and that dye will end up in every progenitor cell after that. Progenitor? No, progenitor is the first one. Every subsequent cell, anyway, will end up with that dye in it. This is just an example of the dye. This is an example of how you inject. So you inject one cell, you let it develop, and then you say, where did the dye end up? Ah, the dye is all in the neural tube. So this must end up being neurotube, neural DNA in the end. It's ectoderm here. It becomes neural tissue in the end. If you inject this one, you might find out it becomes epidermis. You know, these are all epidermis, only this one's neural. Okay, so now if you learn something about the development of the Xenopus. This, by the way, let me blow this up. I used to do these experiments. I'll talk about them more later. But the Xenopus embryo is about maybe as big as the period on a piece of paper. And so you've got these little embryos down there, and you've got this, what do you call it? A glass pipette that you stretch. You know, when you heat glass, it stretches out instead of breaks. So you stretch that glass out and then break it off. And now you've got a tiny, tiny little needle. And you put that needle onto this apparatus and you suck up your solution into that little tiny needle through this vacuum thing here. Okay, you suck up your solution. You go down with this demultiplier apparatus here. So this thing, it's a demultiplier, which means every big move you make with your hand is really small with the needle. So it's decreasing how fast your hand moves. So with this, you can get down in and poke one cell, hit the button, and for a little air to go through this tube here, and then boom, inject that cell. And actually, this demultiplier thing, I'm not sure anybody uses it anymore. Most people will get a mechanical thing. So it's got X, Y, and Z coordinates, and you can just like a video game, direct your needle down there, because these things are really, really tiny. Your hand, and if you drink coffee like me, your hand's doing this, and you're killing embryos. So that's how you inject. As I say, I did that in my first postdoc with Xenopus. It's really easy with Xenopus because, you know, they're fertilized outside the mother. So you just rip that vitelline membrane off, and now you've got access to all the cells. It's easy to inject them. Okay. So fate mapping. So you inject one cell, you find out where those cells go. This is your fluorescein dye. You see it's a huge molecule, just sugar, sugar moieties, what do you call it? Polymer, sugar polymer with the big fluorescent group on it there. So here's an example of fate mapping from your book. So you inject C3, and this is where those cells end up. Here's the dorsal side, ventral side. It's on the dorsal, it's on the right side, it ends up on the right side of the embryo. So this ends up being the right side. So you're going to gastrulate this way. I don't know, so you must be gastrulating this way. Okay. Anywho, here's another example. Now here, in injecting this RNA, we're not injecting something that we can follow. We're injecting the messenger RNA for a protein. So we're going to make the protein, find out what that protein does. So in this case, they've taken an embryo at the 4-cell stage, and they know that this protein beta-catenin comes up on the dorsal side. So let's see what happens if we inject some on the ventral side. So now you're going to have an embryo developing that's got beta-catenin on either side of it. And what happens, this is normal, is you get two axes. So it neurulates in two different ways, gastrulates and neurulates in two different ways, and you end up with two axes. So that shows you that this beta-catenin is really important for producing that axis. And we'll talk about that next week in chapter 4. that's another thing we can do experimentally. Very easy to do. Okay, another thing, which I'm not sure, see, your book does a lot of discussion of the historical experiments. I'm not sure people still do these kinds of experiments, but we will talk about them later in the class. So this is producing a chimera. So two different ways you can do this. One thing people like to do is they'll take chick embryos and quail embryos. So they're different species, different genus, but they're similar enough that quail cells will not be rejected by the chick embryo. So you can take quail embryonic stem cells, from the early blastocyst, blastoderm, and put them into a chick embryo and let it develop. And then you can always find those quail embryos because immunologically they're a little bit different, and you can detect them with like a immunohistochemistry or something. So here's an example of that. This is a figure from your book where, if you can imagine, there's cells all the way through here, but you can only see the ones that are lighting up in red, right? So those are the quail cells. And the clear area, there are actually cells there, but they're chick cells, and you can't see them. So this chimera is a way of putting two different types of cells together, and you can always detect where they are. Here is, again, you've got a mouse at this blastocyst stage. You take cells from a different mouse and you put them in, and early embryo stuff, they're all so plastic. They can change, they can absorb other cells, they can do all kinds of crazy stuff that adult cells wouldn't do. So you add these embryonic stem cells to these other embryonic stem cells, and they just kind of get absorbed into it, become part of that mouse, and you can see spots where the other mouse, the other mouse's DNA ends up. So you've really got this particular mouse has got two completely different genomes. It's got one from a white mouse and one from a brown mouse, and you can see where they ended up. And then you can take this chimeric guy, and you can find out from the sperm and egg where if those cells ended up in the germline, right? Because normally, if you're adding cells together and they end up in the somatic, they become somatic cells. That's it. That's the end of the line. When that animal dies, you'll lose your chimera. If some of those cells end up in germline cells, then you can breed those and then develop a new strain of mouse that only has that new gene that you wanted in there. So chimeras are another way to do that. And again, I'm not sure how much people do this anymore because there's better techniques today. Okay, here's another example. Let's see, do I have it here? Nope, that's the next one. Okay, I'm going to change my share and show you my notes real quick. Come on, you. Okay, so I don't know if any of you have had Dr. Parker at all. He's a big fan of the Drosophila model. He's done a lot of Drosophila stuff. And a lot of really important cell biology has been done with Drosophila, kind of back-asswards, in my opinion. But they found out a lot of really important things using Drosophila. And this is the way they do it. They take a population of them, eggs or fertilized eggs or something, hit them all with a mutagen, like hit them with X-rays or hit them with a chemical mutagen or something. And you do the X-rays or the chemical at a concentration that about every embryo will get about one mutation. You could crank up the X-rays and just kill everything, or have it too high and get several mutations per embryo, or you can just work out the details so you get about one mutation per embryo, and then you let all those embryos grow, and you look at each of the adults, and you see, are there any phenotypic changes that happen in some of them? The overwhelming majority, you won't see anything interesting, but you may see one that's got an antenna coming out of a leg coming out of its head instead of an antenna. Something's going on there. Take that one out and breed it and make more, and then find out what changed. What was it? What protein changed that caused that developmental shift? And so an awful lot of things were learned that way. And I always complain about this in cell biology because proteins are always named something stupid when it's cellular, because this is the way they found it out. They didn't know what the protein did. So basically, you find you mutate a whole bunch of organisms and then let them grow, and you find one fly has got a forked head, you know? Well, that's kind of an interesting phenotype. You take that out, you analyze it, and you call the gene that produced that phenotype forkhead. And eventually you find out what protein makes forkhead, and then you find out it's some kind of signal transduction molecule. So the name forkhead has nothing to do with the function of the protein. It just shows you where that was discovered. Wingless is another one that's really important. Wnt is a protein we'll talk about later, a really important diffusible hormone that is really important for development. And they found it here. You know, they had wingless Drosophila, and because they needed that hormone. Sevenless is like a growth factor receptor. Son of sevenless is a protein associated with the growth factors, son of sevenless. Hedgehog is another hormone that we'll talk about in this class, really important for development. Sonic hedgehog is associated with hedgehog, but also an important function. So I throw that out here because I think it's funny. When I was working at Harvard on my postdoc, I knew a guy in a lab, and that guy's partner they worked on chick embryo development. And a friend of his in the lab is the guy who named Sonic Hedgehog. And he named it that because he knew it was associated with hedgehog, and his son liked to play Sonic Hedgehog on the the console. So he used the name Sonic Hedgehog. So we'll see Sonic later in the class. Just a brush with fame there of mine. I also met Pete Rose for another brush with fame. Okay, and Willie Mays. I met Willie Mays when he was alive. Okay, one problem with this method is it's kind of hard to find embryonic lethal genes. So if there's a gene that's really, really important during embryonic development, the embryo just doesn't develop, and then you don't get anything. So you can't wait for the adult to look at phenotypic changes because you don't get any adults. So it's kind of hard sometimes. And those are the most important genes for development, are the ones that the organism doesn't develop without it. So that's forward genetics. That's one type of method. The other is reverse genetics, which is like you've got a protein you're interested in. Now let's go back and add it to different cells or into the whole organism and find out what it does actually. So this is kind of what I was doing up above when I was talking about, change my share. Okay, so when I was talking about this is the forward genetic route, which is, you want to know what this protein does, inject it in a cell and find out what the function is, find out what the phenotype is after you inject that. But in this case, we're talking about putting it into the genome. So transgenic animals, you take a gene from one animal or even a gene from the same animal but mutated, and then put it into a new one. Whenever you look up transgenic animals, I think it's kind of funny. I think in the popular imagination, this is what we do in the lab. We make things that glow. Because whenever you look up transgenic animals, you see these mice with feet that glow. Chicks, mice with glowing eyes, and of course everyone's favorite, the glowing bunny, which I think you can buy. I think they're available in pet stores. So it's just easy to find them at night, basically. But a lot more important things are done with transgenics than that, and that's kind of one of those funny things that people get an idea in their mind that this is what we're doing, but it's not really what we're doing. This is how it happens. You take cells and you grow them in culture, and then you add a new gene to one of those cells, and then you take the nucleus out of an oocyte and you add your nucleus to it, and activate it. So this is the way they made clones of cloned animals originally. So you're taking your nucleus from a regular cell and replacing an oocyte nucleus with the nucleus from your regular cell, and then activating the cell so it thinks it's been fertilized, and then it develops. And you put it into some surrogate mother, and then the offspring you get will have that new gene in it. So, share my notes again for a second. Sharon is Karen. Okay. Transgenic animals. Okay, so number of different ways you can do this for reverse genetic. Usually it's transient, which means it's only going to affect that one cell or that one organism you're working with. And when the organism dies, it's gone. That's because it doesn't enter the genome. Maybe you're just adding RNA, so you're getting the protein. You could add a plasmid with the DNA in it. It could insert itself into the genome, but not in germline. So you just have this one organism that you work with. The advantage of this is it's really quick and easy. I mean, you could do this. Well, back in the day, we would get embryos we would get eggs in the morning, make embryos. By afternoon, we would inject messenger RNA, and the next day we could do experiments. That's how fast it is. Versus transgenic takes weeks to months before you get an organism. So if you want to change up your RNA to try a lot of different things, this is a good way to do it. So that's the advantage of this. And you can use this for fate mapping too. I don't know, actually, this doesn't belong here. Okay, so, I see why I put this here. So you could put the mRNA for the green fluorescent protein in this and watch where that cell goes through the fluorescence. Or more importantly, some protein like a transcription factor. What happens in that other example where we put beta-catenin or brachyuria or one of these important transcription factors into a cell where it isn't normally, what happens to that cell? And that tells you what the protein does. Okay, but transgenic animals, though, is when you actually put it into the genome and in the germline. So you start out with a single cell that's got a single genome, and you put your gene into that genome. Now, when that develops, the germline cells are going to have it in it too. So now you've got a whole line of organisms that have your gene in them. Old style, the way it was originally done was just random. You put your plasmid to the nucleus of that donor cell, and if you're lucky, it will randomly insert into the genome somewhere. You don't know where it's going to happen. It may knock out another gene accidentally. Odds are it's not going to, though, because as you know, like 95% of your genome is not coding or cis factors. So there's a good chance it's going to fall into somewhere that was not busy anyway, and make your protein. So let's see, gene gets incorporated into the genome somewhere. Inserted RNA could be, so then what is the good of that? Well, you've got your gene, and you could put your gene in that plasmid with a cis region that's tissue specific. So say you wanted to know when does development of the heart start and where. So you could put a green fluorescent protein on the cis region from a cardiac specific gene, put that into transgenics and watch the embryos. So when that gene gets turned on, green fluorescent protein will come in that cell as well. And you can follow the gene expression by following that reporter gene. That's a good word, reporter gene, remember that one. Or you could put, your gene in it on a specific, with a specific promoter you can control. There's a lot of promoters that are turned on by antibiotics. So you put your gene into this plasmid with a antibiotic controlled promoter, you make a transgenic animal, and whenever you give that animal or embryo the antibiotic, that gene gets turned on. So you've got really good control over that expression of that gene. You could put tissue-specific promoters on it, again, so that you can see what happens when this gene gets turned on in this type of cell. The gene you're turning on could be antisense to some protein you want to knock out. What happens if a muscle cell doesn't have MyoD? Well, you put antisense gene in on a muscle-specific promoter and then make a transgenic animal and knock out MyoD. What happens? How does it change the animals? What else? Oh, you could overexpress a protein. You could mutate that protein to be constitutively active, or you can mutate it that it's going to shut off the endogenous protein, find out what happens without that protein. So a lot of different experiments you can do with this technology. One thing that makes the technology, this is pretty much the next step in this technology, is instead of randomly putting it somewhere in the genome, use homologous recombination to put it exactly where you want to put it. So instead of just putting the gene in somewhere and seeing what happens, knock out the good gene and put the new gene in its place. So you've got a gene you really want to follow, find out what that gene does, put your gene in with a controllable promoter, an antibiotic controlled promoter. You can turn that gene on or off whenever you want. And at the same time, you don't have the endogenous gene, the normal gene there, complicating your results. It just makes for a much better experiment. And this is the method that's used. You can look this up. It's in your book. It's also in Wikipedia. I don't want to get into the methodology too much. Basically, you take these lock sequences and you put them at either end of the gene you're interested in, and then you make transgenic animals with that. And then you have other animals that have this Cree protein that will go in and cut out that segment. Blah blah blah. Not that important because it's complicated, and the new method is much easier. So the new method, which you've probably heard about, is CRISPR. CRISPR's kind of revolutionized a lot of, pretty much all of transgenics at this point. So it's based on, let me see if I can pull this up. I don't remember what figure I put here. CRISPR-Cas9. Oh, here it is. Okay. Okay, so CRISPR is based on two things. One is the Cas9 enzyme, which is an enzyme that will have an RNA in it, and this RNA directs the enzyme where to go. So normally this segment of the RNA is for a virus. So Cas9 has this so it can go out and find viruses and destroy them. You probably know how bacteriophage work. They land on the surface of the prokaryotic cell, inject their genome, and start making their proteins. Well, this is going to go and find that viral DNA and destroy it. So it's like an immune response. So that's what Cas9 does. And this protein, this messenger RNA is targeting that Cas9 where to go. So the way it's been adapted for our use is you've got this sequence here, which is the hairpin loop here to fit into the Cas9. Now this sequence, instead of being viral DNA, is DNA from the genome of your model organism. So if you want to change a mouse gene, you put the sequence from that mouse gene here. Cas9 then is going to go into the cell and find that spot and cut it. Now you've got a double-stranded break right there. So once you've opened that up, there's two different things you can do. You can just let the natural DNA repair mechanisms seal that back up. When it does that resealing, most of the time it's going to add or subtract a base or two, throw off the reading frame and screw up that protein. So if you're doing this, basically you're knocking a gene out. On the other hand, you could also include some DNA that you want to replace that other gene with. And so that's homology directed repair, which is another cellular mechanism. If you've got your DNA of the new gene in with your Cas9 and your CRISPR mRNA, then the CRISPR-Cas9 will cut the genome, your DNA will go in, and the normal homologous repair enzymes in the cell will put your gene in where that was broken. Okay? So it's like magic. You can change any gene in an organism today, most organisms, I guess. Whenever in science you make some definitive statement, you're wrong somewhere. But basically, you can change any gene in any organism to something else. And that is just a powerful technique, especially for us in embryology, to find out what each protein does in each cell that causes its results. Okay, so that's our technical that's it for our techniques. Let me show you one thing. Now, this is not required for the class, but, and maybe some of you have heard this, but okay, I want to play this for you because I think it's really cool. I promise you're not going to learn anything from it. Wait, pause. Okay. CRISPR-Cas9. Okay, I had to, oh, stop already. Escape. I had to turn off my video there so you can see me smiling like an idiot. I love that. I absolutely love that. Okay, that's all we have for today. Most of you are gone already, so I will see you again on Monday. Bye-bye.